All right. Hi, and welcome to the next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. Next slide, please. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all our ASPB members who are attending today. Your ASPB membership dues help support making this webinar possible. For any of you who have not yet joined ASPB, you can join today using the discount code %10, where you can receive 10% discount on your membership when you join. As ASPB members, you get access to this webinar and many other ASPB resources, and you can learn more about all of the wonderful things that ASPB does and all the opportunities we provide for the community at aspb.org. Next slide, please. This webinar is recorded, and so we have other Plantae webinars that we've done in the past. So if you're looking for awesome plant science related content, you could check out our Plantae YouTube channel and explore all that we have done so far. Next slide, please. Before we get started, I'd like to highlight a few housekeeping items to make sure that you get the most of your attendance of today's webinar. First, if you're experiencing technical issues, let us know about those using the Q&A box, or you can email me directly at jpadilla@aspb.org. If you have questions for our guest speakers, please also add them in the Q&A box. Our moderator will be sharing them with our presenter during the Q&A portion of this webinar. But throughout the webinar, please, as you hear topics or subjects or things that you might have questions about, please continue to type them in a Q&A. If you are having trouble and need to leave the webinar early, again, we are recording this webinar and will be made available in a YouTube channel. So be sure to follow our YouTube channel so you can also be in the know of when the new content are posted. And so without further ado, I am now going to turn this over to our Plantae Fellows who are moderating today's webinar and facilitating this conversation. And so with that, take it away, Plantae Fellows.
Thank you, Jason. And hello, everyone, and welcome to the Plantae webinar, a smaller carbon footprint in plant sciences. We are very glad to have such a wonderful audience joining us from across different regions and different time zones. So we all know that advances in plant sciences have greatly accelerated research, but they also come with increasing environmental cost, like from energy intensive laboratories to computing infrastructures to large-scale field experiments and global research networks. So our science has a footprint, right? So today we ask, how do we fix that? And so before beginning into the webinar, let me briefly walk you through the outline of our webinar. So we will begin with the introduction of speakers and moderators, and this will be followed by the speaker presentations. And after the talk, we will move into the panel discussion and Q&A session, and where we encourage all of you to please participate by sharing your questions and thoughts. We hope that this session will spark ideas, conversations, and action towards building more environmentally responsible research practices in our community. So today, our three speakers are going to take us from genes to the soil to the choices that we make in everyday science. Firstly, Professor K.C. Bansal will discuss how genome editing can help to develop crops that use fewer inputs and store more carbon. Next, Dr. Yadunath Bajgai will highlight sustainable land management practices such as no-till farming and other to improve the soil carbon storage. But unfortunately, our third speaker is not able to join us due to some medical emergency. So we will go ahead with the talks, and we hope that today's discussion will gain insights on how research, innovation, and the responsible practices can contribute to building a more climate-conscious future in our field. So today's webinar will be moderated by me, Sonia Balyan, along with other Plantae fellows, Kavita Joshi and Trevor Melusin. So, just a brief introduction about myself. I'm currently working as a scientist at Indian Biological Data Center, Faridabad, where I lead the development of uh, national data resources. And apart from that, I am also the founder and host of Beyond Shod podcast, and I'm also associated with the International Society for Biocuration. So now I invite Trevor to introduce himself. Trevor? Go ahead.
Thank you. My name is Trevor Mellison. I'm currently a researcher at Plasmidsaurus. I work with next generation sequencing, and I'm excited to find the intersection between that and plant science. And I'm very excited to see how this webinar goes and to hear from our speakers. Next, we're going to have Kavita Joshi.
Hi everyone, I'm Kavita, Plantae Fellow 2026, based on Omaha, Nebraska. I'm passionate about plant sciences and enjoy making it simple and engaging for the others. And currently I'm here and actively exploring research opportunities in United States. Thank you.
So now I invite Kavita to introduce Professor Casey Bansal. Kavita, please go ahead.
Thank you, Dr. Sonia. So our first speaker for today is Professor Casey Bansal. Professor Bansal is a plant biotechnologist with expertise in functional genomics, genetic engineering, and genome editing, aimed at developing climate-resilient crop and sustainable agriculture system. He earned his PhD with a gold medal from Indian Agriculture Research Institute, New Delhi, India, and later did postdoc at Harvard. He later served as a visiting scientist at Rutgers University under the Rockefeller Career Biotech Fellowship in 1996. Professor Bansal served as a director of premier ICR Institute like NBPGR, New Delhi from 2010 to 2016. He represented Asia as a vice chair genetic during the 15th regular session of the United Nations Commission on Genetic Resources for Food and Agriculture from 2013 to 2015. He also served as a secretary of the NAS India from 2021 to 2023. He has authored over 150 research publications in leading journals such as Nature Biotech, Nature Genetics, Nature Plants, Nature Communication, Trend in Plant Science, Plant Biotech journals. Professor Bansal has received many prestigious awards, including the National Award from Indian Science Congress, given by the Prime Minister of India, and the Haryana Vigyan Ratna, the state highest science award. So, more recently, he was elected a Fellow of the World Academy of Sciences for the year 2025. So far, only 8 Indian scientists have received this international distinction in agriculture, nutrition, and food system sciences. In addition, Professor Bansal received the prestigious Dr. B.P. Pal Memorial Lecture Award in 2025 by the National Academy of Science, India. So it is a privilege for us to have you with us, Dr. Bansal. So over to you, sir.
So now,
Okay, thank you. Can I start sharing my slides, please?
Yes, sir.
yes.
Yeah, thank you very much, Kavita, for your kind words of introduction. And I'm pleased to be here, speaking today on this particular topic, that is lowering the carbon footprint in plant research. Give me a minute for sharing my slides, please.
Sure, sir.
Are you able to see my slides, please?
Yes, sir.
Okay, so I invite you all to this particular webinar and to this particular topic, that is CRISPR-based genome editing for carbon efficient crop improvement. And as you know that the whole webinar concept is on reducing the carbon footprint in plant science research. But again, the question is, why do we need to reduce carbon footprint? Because to combat global warming has become a big issue, and the rise of carbon dioxide concentration in the atmosphere, is causing this global warming, and which in turn can reduce the frequency and intensity of climate-driven disasters like, extreme temperature, floods, etc. And as a result of this buildup of, all these gases, you can see here the number of natural disaster events, they're increasing right from 1970 for the last about 55 years. You can see the number, be it kind of high temperature, extreme events, or floods or droughts. They are on a rise, comparatively speaking, in the last about 50 to 60 years. But again, then how can we reduce our carbon footprint through plant science research? There are various ways. One, as was introduced by Roxonia, that's the one. Otherwise, also we can help reduce carbon footprints by enhancing natural carbon sequestration in plants, and by therefore developing climate resilient crops. And if I put it in perspective with regard to the use of CRISPR genome editing to develop climate resilient crops, as Dr. Jennifer Doudna, the Nobel laureate herself, talks about, that we should try to develop crops with enhanced heat and drought tolerance and water conservation, so that there's more retention of water in the leaves rather than it get transpired. And engineering microbes even to curtail dependence on synthetic fertilizers, because that's also, of course, in relation with the plant growth and development. Crops to fix more carbon dioxide for increased carbon sequestration in the soils. And also it's important to reduce greenhouse gas emissions from agriculture, particularly as we know the experience, or even globally, from rice fields, and minimizing farming inputs, because as we can reduce the use of fertilizers or water, also can reduce the greenhouse gas emissions. And of course, this is a general message which is quite encouraging, from Dr. Jennifer Doudna, that CRISPR-based solutions will allow farmers to adapt to a changing climate and sequester more carbon while preserving prized regional varieties. And I will talk to you a little bit about that as we go further. And she further said that there's an incredibly important role for this particular technology, CRISPR, in the protection of small farmers. So in India itself, we are working on climate resilience, through genome editing tools. And we, of course, trying to incorporate all those relates related to climate resilience, including, increased radiation use and photosynthetic efficiency, which is very important to capture more and more carbon, sequester, deeper into the, soil, and increase the organic structure. In fact, in August 2024, our Honorable Prime Minister of India announced a policy, we call it as Bio-E3 policy, that is biotechnology use for economy, environment, and employment. And out of 6 thematic sectors, one of them is on climate resilient agriculture, and with a goal to contributing to India's net zero emissions by 2070. And we also making all of our efforts on use of this technology in about 40 crops across 34 institutions of Indian Council of Agricultural Research, primarily with a focus on climate resilience and sustainable agriculture. But then if you look into different strategies for carbon efficient crops, there are several of them. To start with, more important is enhancing photosynthesis. World over, work has been going on, and on engineering the C4 traits in C3 plants, for example, particularly in rice, and which has been a long-standing interest for the last about 15 years. People have been working on it in different, international consortia, including, International Rice Research Institute in Manila. The next is, can we increase nitrogen use efficiency? Because nitrogen use efficiency remains very low when we talk of utilization of it by plants, remains only about 30 to 40%, or even up to maximum 50%. So can we increase this so as to reduce the use of fertilizers, which can in turn reduce the emissions of methane and, more importantly, nitrous oxide, which is also contributing to global warming, along with methane. Then the third strategy is, can we reduce stomatal density, or can we have stomatal regulation for opening and closing, for improved water use efficiency or reduced transpirational losses of water? Next is root system engineering, which is a little bit, in fact, of quite concern, because there's not really much effort into it. We need deeper root system. Again, there is a discussion going on that how much deeper root system will really help when we want also more shoots and more biomass for increasing more yield. Nevertheless, deeper root system for more carbon storage in soil is very, very important. So CRISPR, I said before, is being explored globally to enhance carbon capture in crops. And just to inform the students, that, there is a story going on in the world I mentioned to you about, like for the last 15 years, to develop C4 rice. Rice is a C3 plant, maize, sorghum, sugarcane, they're all C4 plants. C3 plants actually have only one single type of photosynthetic cells, that is mesophyll cells, whereas in C4 you got two different types of cells. You can see here, these are bundle sheath cells, and around these bundle sheath cells we have got mesophyll cells. And these, therefore, photosynthetic activity in enzymes, they have got a kind of a division of labor. But initially, these are the four enzymes which are very important. The first one is phosphoenol pyruvate carboxylase. And in fact, when we talk of installing these biochemical and anatomical attributes, which is necessary to support C4 photosynthesis, this work is going on in C3 crop rice for quite some time. While all these enzymes have been overexpressed, but there are issues still for anatomical attributes to be located in C3 rice, in C3 plants, particularly because you need chloroplasts, which are dimorphic, both in mesophyll cells or also in chloroplast cells. While in rice, this is of course a transverse section of maize, but if you see in rice, you don't see any chloroplasts actually in bundle sheath cells. So there's a need now fine-tuning with rapid advances in crop gene editing to ensure that we do get success in developing C4 rice with the help of CRISPR-based genome editing technology. But in the meanwhile, one of our scientists in India is using the C4, is generating C4 like PEP carboxylase in rice using precision genome editing tool. In fact, if you compare only the PEP carboxylase, I'm talking about it here, in these C4 plants versus C3 plants, there's a single amino acid change, from glycine to arginine. Or you can say here in C3 you got arginine, whereas in C4 you got glycine. And it has been determined before, that this is a single nucleotide change which actually, dictate in a way the malate inhibition. There's more inhibition here in case of C3 plants and less inhibition or no inhibition in case of C4 plants because of the single mutation. So Dr. Kutubuddin Molla from one of the institutions on Rice Research Institute in India has taken up this work to change CGC, which is a codon here, making arginine in C3 plants, to GGC, which will make glycine. So if you convert C to G, then GGC will be, encoding for glycine. And with the presence of this glycine now disposition will have increased tolerance of this particular enzyme, PEP carboxylase, towards, feedback inhibition. So feedback inhibition, is the one which is otherwise lowering down the activity, and that will be released if this particular mutation is brought in. So he did it in rice, and the edited lines accumulated more chlorophyll and also showed more photosynthetic activity. You can see here clearly the wild type versus the edited line, and when you see the chlorophyll content, in terms of milligram per gram fresh weight, you can see here is less than 1 milligram, but whereas it is touching even most of the edited lines up to 2 milligram. So there's a 40 to 50% increase, and that is translated into increased photosynthetic rate in terms of micromole carbon dioxide per meter square per second, which is normally about 20 to 25 in wild types in C3 plants, including rice, wheat, even others. But here in edited lines with only single amino acid change in PEP carboxylase, you can see here the activity has gone up to even 30, micromole per micromole carbon dioxide per meter square per second. So there's about 40 to 50% increase, which is quite encouraging. Though I must also say here that this PEP carboxylase, which is edited, is not really playing any direct role in C4 pathway, while it is the first enzyme required. But here he discovered it as having more kind of iron uptake. And because of that better iron uptake, though he's studying the mechanism, there's more chlorophyll synthesis, and edited lines has more chlorophyll. And as a result of more chlorophyll, of course, there's already a relationship established. It has more photosynthesis. So, but I actually firmly believe that C4, it may not be, to me, it appears in the long drawn procedure, to convert C3 into a C4 rice. But in the meanwhile, we must also look into some of the wild species of wheat, or for that matter, even rice. There's a lot of genetic variation. This is my own work when we have seen a lot of genetic variation when you talk of, diploid or tetraploid or hexaploid wheat. Hexaploid wheat or some of the tetraploids have got even dicoccum or urartu. They have got very high rate of photosynthesis. There's a lot of genetic variation demonstrating a greater diversity, number one, and also superior leaf photosynthesis in comparison to cultivated elite lines. So I'm talking, can we now, with the availability of CRISPR, go for neo-domestication, of C3 wheat with higher rate of photosynthesis, with higher capturing capacity? while the rice, of course, the working going on and C4 rice, but rice also, it has been demonstrated that neo-domestication of the wild species is possible, and we can try to, in fact, incorporate, better rate of photosynthesis. Also, CIMMYT believes that some of the ancient wild relatives, they hold key to climate proofing because of the high rate of photosynthesis. And this is one of the recent papers where it also says there's a lot of variation in leaf photosynthetic traits across species. These are different species. This is some of the diploids, tetraploids, and hexaploid. And this is the rate of photosynthesis. Rate of photosynthesis, again, micromole, carbon dioxide fixed per meter square per second. Here it goes up to 60. Normally, I said in case of C3 plants, I mentioned before, it's about 25 to 30. But you can see here, even in hexaploid, while they already known, these diploids have got a high rate of photosynthesis, or tetraploids, but even diploids, even hexaploids, which is a bread wheat, which is cultivated by the farmers, even it has got also a lot of genetic variation. And you can see here, it has got higher values up to even 50 micromole compared to about 25 to 30. So this could be, in fact, also taken up as a transformation through genome editing, some of those species, and retaining all that high photosynthetic rate, but adding other traits which are important, for popularization in farmers' field, some of the agronomic traits. So next is increased nitrogen use efficiency and stomatal regulation, or reducing stomatal density. I will take it together, through the use of CRISPR. That's work which has been already done in India in one of the projects which we formulated about 4 years ago. And these lines have gone into trials already. One of them is the DHT mutant, which is a zinc finger transcription factor, which controls the expression of several genes, and which is also responsible for not only giving tolerance to drought and salinity, but also it is increasing. You can see grain yield up to 20-21%, and this has also got wider or broader leaves with reduced stomatal density, and therefore it has got the capacity to enhance water retention under drought stress conditions. Similarly, the next one we have is another line, genome edited, with the cytokinin oxidase gene, which has got also a higher number of grains, higher more number of spikelets, and overall these two varieties have been, of course, recently launched just a year in May, on May 4, 2025, by actually some of our ministers of agriculture, and you can see here these two lines. This particular line, DST rice muted line, has got up to 30% more yield in saline soils. And this is, again, it has been mentioned and studied. There's some of the genes which are responsible for it. That's due to the downregulation of stomatal developmental genes, which are again transcription factor genes. So as a result of that, there's a reduction in stomatal density. I mentioned before, there's a reduced stomatal density. The other one is also this particular line with edited of CKX2 gene in rice. It matures 20 days earlier and saves water and fertilizer. So if it saves water and fertilizer, though it is not directly playing any role, this particular gene, that it except it matures early. But when you save water and fertilizers, it will, of course, cut down on the emissions drastically when we talk particularly the nitrous oxide or even methane from rice fields. And as you can see here, there's a 20% lower greenhouse gas emissions, while there's a 19% more yield. Together, we talk about it, these two varieties, and 7,500 million cubic meter of water is also saved. So which is, in fact, a direct role of CRISPR through particular genome edited of rice varieties, which we observed in India. And the last category I'm going to talk about briefly is the root system engineering. But root system engineering, actually speaking, while we do need deeper roots, but then we need to screen germplasm to have these deeper roots, and without any kind of a reduction on the biomass, which is very important. So therefore, the partitioning has to be really studied well here. But again, not only for this particular trait, but even for all other traits, we need to screen the germplasm. And screening germplasm is very important. And why it is for important? Because there is a limitation and a challenge. There's a lack of knowledge on suitable target genes when we talk of the other traits which I've already mentioned, though which are already edited, some of the genes. But then the identification of these target genes and alleles, particularly in some of those plant genetic resources or wild species for the desired traits, is essential. And we have in India 0.5 million accessions conserved. We are using it. We are actually using it all, characterizing it, screening it against different stresses. Here, of course, I'm giving more emphasis for this particular webinar on our results on terminal heat stress tolerance. About 22,000 accessions, we screened it under hotspots, and you can see a lot of variation. it covers the ground early, saves a lot of moisture. It reflects the radiation back, because of its, this kind of a color or waxy nature of the leaves. So there's a lot of variation which we found as new sources for terminal heat stress tolerance. Out of 20,000, we got about 3,000 wheat accessions, depending upon all these traits which are related to, high temperature tolerance, particularly in wheat. And similarly, we, also characterize entirely the chickpea germplasm, about 14,500, because chickpea and wheat are two very important crops when we talk of nutrition food security. And this is, of course, done in collaboration together with scientists from NBPGR. So important message for identifying some of the genes today we are talking about is the use of pangenomes or super pangenome, when we can have the genome sequencing of thousands of species, not only a single reference genome. And as a result of that, you can identify new gene families. You can identify new dispensable genes which are responsible, actually speaking, providing tolerance to various kinds of abiotic stresses, for even higher capturing carbon capacity, or for increasing nitrogen use efficiency, water use efficiency. And this kind of a variation will encourage the use of some of those wild species as well. This is an example I'm giving you of chickpea sicer species, and which could be then later used not only through marker-assisted selection, but also for genome editing. Because genome editing, as you know, is catching up, wherein precise modifications you can do, and I can talk to you more about it if required in our Q&A session. So finally, what I want to convey a message here is that you need to integrate gene bank collections with genome editing, not only for carbon capture, for all other traits, and finally, you install the genetic variation through genome editing in elite crop varieties popular with the farmers. And in India, these varieties, improved varieties, through genome editing, are exempt from biosafety regulation. I can talk more about it if somebody has some questions, and I'll be happy to take care of all this. With this, I thank my collaborators, Dr. Vishwanathan, whose work I showed you on DST1, Dr. Rajiv Varshney, who's working collaborating on genome sequencing, Dr. Kutub Mola's work, and I thank scientists of NBPG, our entire collection of chickpea and wheat we characterize together, all these agencies. And of course, I'm thankful to Dr. Jennifer Doudna, with whom I had an important and, actually speaking, a pleasing opportunity to meet her in January 2024 when I visited University of California, and I was there in her institution for some time. So thank you all, and I'll be very happy to take up some questions at the end whenever is required. Thank you again, Jason. Thank you for this opportunity.
Thank you, Professor Bansal, for your insightful talk. So I now hand over to Trevor to introduce our next speaker.
Thank you. I'm honored to introduce our second speaker today, who's going to be Dr. Yadunath Bajgai. Dr. Bajgai is a research scientist at Ohio State University's Ratan Lai Center for Carbon Management and Sequestration. Originally from Bhutan, he built his academic foundation in the UK, Thailand, and Australia, earning a PhD in soil science from the University of New England in Australia in 2013. He spent over 2 decades working in Bhutan's agricultural sector, first as an extension and district agriculture officer, then as a researcher focused on soil fertility, sustainable land use, and potato breeding. Notably, he led work that resulted in 3 different potato varieties being officially recognized in Bhutan, and he also initiated a major 5-year JICA-funded research project. With more than 60 publications to his name, he now works at OSU on the Sea Farm project under Professor Ratan Lai, focusing on carbon sequestration in croplands, rainlands, and grasslands, with the dual goals of improving soil health and helping mitigate climate change. Thank you.
So, Dr. Bajgai, you can now share your screen, or you want us to run the slides?
Let me try sharing from here, so it's easy to click.
Yes.
It has come, but I'm trying to make it full screen.
I think if you go to slideshow, you should be able to click present on the options up above.
It's here in the bottom.
Yeah,
In
okay.
the
Yeah. Can you see now the full slide?
Yes.
Okay, so it's my turn.
Yes, please.
Thank you very much, Professor Bansal, for providing this wonderful presentation on how can we generate carbon efficient crops. And I would like to take forward to help how can we retain this sequester carbon in the soil. So I think this is really wonderful, and I would like to thank everyone for joining and host. Today, my topic is on soil and plant centers and strategies for reducing carbon footprint. And as you can see, this presentation is co-authored by my supervisor, Professor Ratan Lal. So, what are the pathways that transfer carbon dioxide from atmosphere to plant and soil? Carbon dioxide captured through photosynthesis is stored in plant biomass that we have seen in the previous presentation. Plant and crop residue, dead residues, return this carbon to soil. Root exudates supply labile carbon to soil microorganism for decomposition. This decomposition of plant and animal input transforms carbon into labile and stable pools. A portion of this carbon retained is in the soil is as soil organic carbon, as you can see in a picture. So in this context, what do we mean by smaller carbon footprint? In plant science, or in its applied form, which is agriculture, it includes both sources and sinks. Sources include carbon dioxide, nitrous oxide, methane from fertilizers, soils, machineries, livestock, etc. Sinks include CO2 captured by photosynthesis and carbon stored in plant biomass and soil organic carbon. Therefore, a smaller carbon footprint means lower emissions per unit of biomass or yield produce, or greater carbon retention in soils and plants. That is, net greenhouse gas emissions associated with producing biomass or food is lower, and it is expressed as carbon dioxide equivalent because nitrous oxide is 300 times and methane is 30 times more global warming potent compared to carbon dioxide. This is usually measured per unit of production, per kg of grain or plant biomass. So what are our strategies in agriculture? The strategies are conservation agriculture practices. As you can see in this diagram, the strategies are minimum soil disturbance, maximize soil cover, diversified cropping system, pressure on land and soil increases to produce more food. So that is why these three basic conservation agriculture practices have been promoted to protect and restore soil health, which provides ecosystem functions. To sustain and restore soil health, these three principles is adopted. Conservation agriculture practices for soil health. No tillage cropping system. As you can see, corn residues here in the picture. Soybean is growing. It is picture that I took it at Delaware County. Soil disturbance is only restricted to sowing operation in no tillage system. And crop residue retention adds soil organic matter to soil. Other CA practice that is used is cover crops that prevent soil from exposure to heat, protects it against soil erosion, and add organic matter to soil when it is plowed back. Tillage affects soil carbon storage. You can see in this picture from our recent publication, with trees is a native vegetation. In the middle, you can see plow-based tillage. At the left is no-till systems. Native vegetation has soak in equilibrium. Conversion of native vegetation to cropland decreases this carbon storage. Carbon dioxide is released mainly because of breakdown of soil aggregates, where carbon is held. Transition to no-till system demonstrate the soak restoration that you can see in the left. Therefore, long-term no-till is the step forward in reducing CO2 emissions so that we can reduce the overall carbon footprint of producing food. How does no-till management reduces carbon dioxide emission? You can see in the picture that when you plow the land, what happens? It breaks soil aggregates into smaller pieces. In doing so, carbon held in the soil aggregate is released to atmosphere. So no-till farming minimizes soil disturbance. It offers several benefits, including preservation of soil structure for maintaining storm within soil aggregates. It improves soil moisture retention by enhancing water infiltration rate, reducing surface runoff losses, while lowering labor, time, and cost associated with tillage operation. In contrast, conventional tillage involves soil inversion or mixing, which disrupts soil structure and exposes some in aggregates to microbial breakdown, microbial decomposition. For example, shifting from conventional to no-till practices has been shown to decrease runoff by 43% and sediment losses by 87%. That's why no-tillage practices are better than conventional tillage in retaining soil organic carbon. Cover crops. Purpose of planting cover crop is to maintain soil cover during fallow periods to prevent bare soil exposure and reducing the risk of soil degradation. As you can see, as I demonstrate in the picture, cover crops including grains, brassicas, grasses, legumes are sown after harvest in the fall and terminated before planting next crop in the spring in the US. And it depends where we are in the world. Growing mixture of cover crop species has been shown to improve plant productivity, diversify root architecture so that plant reaches different parts of the soil profile and ground cover. The practice offers multiple benefits, including reduced soil erosion, improved weed suppression, decreased soil compaction, and enhanced soil moisture and nutrient levels, increase overall crop productivity. That is what we are after. So another strategy is reducing carbon footprint, integrating livestock with crops. What it does, it recycles plant biomass through livestock because crop residue is either used as feed or bedding material, and it returns nutrients and carbon to soil as manure. It reduces reliance on external inputs like fertilizers, strengthens whole system carbon cycling, improving soil health and fertility, enhancing water retention, reducing soil erosion, and increasing resilience to climate variability. So far, what I have talked is on environmental benefits. Now, can I talk about financial incentives? Why should our farmers do this? Why should they practice this? So carbon, which is a concept called carbon farming, it is a new income stream for farmers. Adopting improved soil management practices can generate carbon credits when they reduce, avoid, or remove greenhouse gas emissions from the atmosphere. For instance, conservation agriculture practices or afforestation can increase soil organic carbon stocks, thereby enhancing CO2 removal or lowering atmospheric concentration of carbon dioxide. To quantify these benefits, soak stock changes from baseline to after adoption of practices needs to be studied. Because carbon credits account for multiple greenhouse gases, information on how overall greenhouse gases fluxes change relative to the baseline is also required. I would like to leave this slide by defining what do we mean by carbon credit. A carbon credit is a tradable certificate that represents a reduction, removal, or avoidance of 1 metric ton of carbon dioxide or its equivalent. So in conclusion, I would like to say that soils and plants are central to reducing agriculture's carbon footprint. As you can see, and we have seen in the previous presentation, that crops and plants have the ability to sequester, take the atmospheric carbon dioxide and lock it up in plant. As you can see, this soybean crop growing in the Midwest, USA. So building soil organic carbon and biotic carbon improves crop productivity and system resilience, which is what we are after. Carbon efficient systems sustain both food security because of enhanced productivity as well as climate goals because atmospheric carbon dioxide can be sequestered in the form of plant biomass, in the form of soil organic carbon, and by different means. Carbon economy. Lal Carbon Center at the Ohio State University offers educational program on soil carbon management and sequestration, and this is offered for graduate and undergrad students, for extension educators, farmers, and general public. I would like to request our viewers to please visit the website that I have presented in the screen for details and upcoming programs. I would like to thank Foundation for Food and Agriculture Research for supporting my work. Thank you to CFAM sponsors for supporting the work. And I would like to thank everyone, hosts, other presenters, and the viewers of this webinar for attending this seminar. I would like to request everyone to make this, take this a challenge and in sequestering atmospheric carbon dioxide in locking up in the soil so that our future generations can have the better earth so that the climate change can be mitigated. Thank you very much.
Thank you, Dr. Bajgai. Next, we will have about 15 minutes for a Q&A session. So if you have any questions for our speakers, you can type them into the Q&A box and we will ask them of the speaker for you. First, we have some questions for Dr. Bansal. The first one is going to be, what are the biggest knowledge gaps in identifying climate resilience target genes today?
Well, the main thing is that we have no data available on characterization and evaluation of some of those germplasm resources which are conserved globally. Only about 2% of the germplasm resources available globally are being used currently by breeders, in use, in their breeding programs. But meanwhile, we have lack of knowledge on the characterization, phenotypic characterization. I'm talking about it, and it's now only, as I mentioned before, pan-genome sequencing is going on, and that could probably add more value into identification of some of those target genes. Because ultimately, it's important not only target genes to identify, but those genes must also have some kind of a correlation with the phenotypic appearance or the trait we are talking about it. So I think while we do have already, I must say that the target genes identified, but again, we want it particularly in a given species, which is important, which we are doing genome editing. And more than identifying the target gene, which could be a structural gene, which is encoding for an enzyme, important is that we don't have much information available with regard to regulation of the gene expression, which again controls particularly some of the multigenic traits like photosynthesis itself. You need to identify, apart from those 4,5C4 enzymes genes, which I mentioned about it, or structural genes, which are key rate limiting enzymes, in the photosynthetic pathway, in the dark reaction or C3 cycle, we talk about it. In fact, the regulation is very, very important. So I'm happy at least to mention here that the Innovative Genomics Institute of Dr. Jennifer Doudna again, Dr. David Sage is working, and they have come out with a recent very good paper in Nature Biotechnology recently, where they've actually created a library of certain, genes, 3 rate limiting genes from sorghum. And that library they've already kind of analyzed it, and where in the promoter region with regard to the regulation within 500 base pairs, and I think the more important is that as they go further, the kind of functional relationship with some of the photosynthesis or other traits, that's more important. Yeah. So it's not that straightforward, but I'm sure things are happening, and we will have some good leads available soon, I hope.
I'm kind of on that same vein. Someone asks, which target genes are currently the most promising for improving carbon sequestration efficiency?
This question for me, you said?
Yeah, sorry.
Yeah. See, as I said before, the 2 types of approaches which are going on currently. One is that you increase the C4 rate of photosynthesis or install it in C3. And other is that we try to capture more carbon dioxide by C3 plant themselves. And instead of identifying the genes, what my hypothesis, which I presented to you today, would be that can we neo-domesticate those wild species, particularly which are tetraploid or hexaploid, because which are cultivable in the farmer's field, while we retain those genes, for high photosynthesis, and we bring in other genes which are required, which they lack, which are now present, for example, resistance to diseases or bigger, leaf or any other, kind of a trait which is required. We can try to bring on those in those wild species already having high rate of photosynthesis. As we know that in case of rice, for example, neo-domestication has already been demonstrated in about half a dozen of crops. Neo-domestication, some of those wild species have already been, demonstrated. But again, I would repeat that instead of identifying the target genes for carbon capturing, more important will be to understand the regulation part of it, because it's a multigenic trait, and we don't have any success yet, even, for example, identifying the key regions of ribosco enzyme itself, which is responsible for increased photosynthesis in plants.
Great.
Over to you, Trevor.
Thank you. For Dr. Bajgai, we have, how can plant breeding support carbon efficient cropping systems alongside soil management?
Yeah, I think this is very useful question, as a very broad, and as I said in my presentation, converting the atmospheric carbon into a plant biomass is one thing, one part of the story, but how to retain that for longer time in the soil or in plant biomass is another part of the story in carbon science. So in farmers field, well, most of where crops are grown, the new technology that we can do is conservation agriculture practices, except for like if you are growing only trees which are not which are harvested after maybe many years, 100 years. For crops, I think we have to try to practice some sort of conservation agriculture practices so that the biotic carbon, biomass carbon that we have sequestered, plants have sequestered, can be locked up in the soil for the long time. So if we add the residue in the soil, but if we keep continuously tilling the soil intensively, what we are doing is we are activating the soil microbes to mineralize already sequestered carbon and also decompose materials. So one of the ways that we can do is follow minimum tillage, no tillage. Also, we can also grow some short cover crops. That means within short time they can grow, and these things can be plowed back into the soil, and something can be added, including nutrient. Thank you.
Great, thank you. For you again, Dr. Bajai, we have a question. Someone says, a lot was spoken about making arable farming more environmentally friendly. Controlled environmental agriculture and hydroponic systems are often praised as being highly efficient, but in practice have many energy demanding aspects. Do you believe these systems have a use, and what do you believe the key points to improve in these practices are?
This is about greenhouses used for producing crop. Can you repeat it, Trevor? Sorry.
Yeah. So they say controlled environmental agriculture and hydroponic systems are often praised as being highly efficient, but have many energy demanding aspects. And so they're asking about the merits of those systems.
All right. Every system has their own merits and demerits. So in terms of carbon sequestration, I think control environment, it is intensive form of cultivation because energy utilized is more, and uh, it has benefits on producing good quality crops, producing in off-season crops, but other demerits like how much carbon it will sequester or whether it is good for uh, sequestering carbon. I need to look at the literature again, but that's what I would think.
Great, thank you. For Dr. Bonsall, we have a question asking which crop traits you believe should be prioritized first if the goal is maximum carbon footprint reduction.
Well, that's a good question. I think I would prioritize, first of all, increasing nitrogen and water use efficiency, two together, before we even try to increase carbon capturing capacity, photosynthesis, which we have not been able to really speaking do much, while a lot of work is going on. But what is more amenable to me, it appears, is that if we can increase the nitrogen use efficiency, and save the use of nitrogen, and manufacturing of nitrogen by itself is contributing a lot to greenhouse gas emissions. So both ways, we save on emissions. So I would keep, fertilizer use efficiency in general, you talk about it, particularly nitrogen use efficiency. Again, in crops, mostly where it's utilized is rice, where it matters most with regard to greenhouse gas emissions. And second would be, of course, for any crop for that matter, increasing water use efficiency. And sometimes we do talk about, tolerance to dehydration stress or drought stress. But water use efficiency, it's a different parameter again, that how much water the leaves can retain and can utilize it whenever it's required by, in fact, regulating the stomatal opening and closing. Because we need to, balance the two, very important, because we don't want to lose, on kind of intake of carbon dioxide for which we need stomata open. But at the same time, we don't want to close it completely to save on transpiration. And also when we talk of climate resilience, both are important, tolerance to drought and heat temperature, high temperature. High temperature stress would require plants to keep their stomata open to have more evaporational cooling possible. And drought stress will tend to keep stomata closed. So what I'm saying is this is a kind of a balance we must strike. But if these two traits can be managed, I think we're doing very well.
Great.
Very good priority. Thank you.
Let's see. Our next question for Dr. Bajgai is how do you picture the carbon credit system being applied at a field level?
Oh, yeah. I think that's a very, very good, very important question. It is being applied in the US based on the market, carbon market is applied. What we need to do is we need to bring our farmers, growers into agreement so that, uh, their carbon, whatever they have sequestered, can be traded. Whoever are emitting greenhouse gases, emitters can buy, and it's possible, and it's done. It's being practiced here. But carbon markets are depending on which market it is. It depends. So yeah, it's possible. it is utilized today, but it is mainly driven by farmers' willingness to trade their carbon. And trading carbon alone is not even good enough because it has terms and conditions to follow the growers. Already sequester carbon will not do good, but we need to sequester, we need to show avoidance, reduce greenhouse gas emissions. So carbon trading is there and it's coming up, carbon farming. So in basic, but also, we need to look at the market again, and I'm not market expert myself. So that's my point.
Great, thank you. Let's see. This question appears to be for both of you guys, and they're asking whether there's specific subfields, institutions, or roles that we can play in plant science where a researcher could have the greatest impact on reducing our carbon footprint.
He's talking about what kind of institutions you mean? What was the initial question, please? Or can you repeat?
Yes, they're asking like if there's any specific career paths, it seems, in plant science, whether that's like a subfield or a role or a specific institution you could be at where you could have the greatest impact.
Yeah, I will take it. In fact, you know what, this is certainly a very important area of research currently available, and in the sense which we must pursue. But I would only like to actually speaking give a message to the students, particularly that we should, try to study the physiological mechanisms, biochemical mechanisms of carbon capturing capacity, how to increase it, or what are the rate limiting enzymes and parameters. Of course, a lot of work is going on, and then you can try to also use, genetic resources, screening them. Already you will have a lot of differences available in terms of various physiological parameters, so irrespective of the enzymes which contribute to this, carbon capturing capacity. So there are very many institutions, working on it. When you talk of India, suppose, then we have Indian Agricultural Research Institute primarily working on it. In fact, there are different divisions, including plant physiology, biochemistry. We have got a very strong department of genetics and plant breeding. They can all, together, we can speaking, make a project and work on it. Though there are other institutions, of course, across the world, but particularly I'm mentioning to you about this institution at the moment, which is quite famous in India.
Thank you. Dr. Bajgaya, do you have anything to say on the subject?
I think that is good enough because I'm more working on soil part of it. So we have to work on institutions that work on breeding, genetic engineering, that aspects, I think. So I think that will suffice. Professor Bansal's comment will suffice that.
Great, thank you. Let's see, let's get one more question. For Dr. Bansal, it looks like, which infrastructure do you see is required to scale precision breeding using CRISPR?
Which structure? What is the question? Sorry, I think I have some.
What infrastructure?
What infrastructure?
Yes.
For increasing precision breeding, for scaling it up, you mean?
Yes, scaling, exactly.
We are talking about speed breeding is one way forward. But to me, it appears that first of all, we must be able to develop the primary genome edited line in a particular crop with a given target gene for a particular trait. And once you have that and you have verified the edits and you have confirmed that there's no foreign DNA present, because that's from the regulatory perspective, it's very important in most of the countries. I mean, it's required even if it is to be declared as non-GMOs. So once you have verified the edits, you have found that there's no foreign DNA present anymore, you can straight away go to the field trials as far as Indian regulatory system is concerned. And if you want to really scale it up, for subsequent generations for stability, kind of analysis, etc., you can go to, speed breeding, and you have to, of course, have to develop a protocol and system, which are there available now, I'm sure. At least in India, we know a few of the institutions are working on it, and you can scale it up. But to me, it appears speed breeding may not be as important. Important would be to create the primary, genome editing event, which is important for a particular target gene. So looking for more target genes, I would give more emphasis than to, of course, scaling it up will come up later through field trials. Even, partnering with private sector is very important in this case. Private seed companies.
All right, thank you both for answering our questions. It looks like we're just about out of time, so I'm going to pass it over to Sonia to finish us off.
Thank you again for very insightful talks. And now we came into this hour asking one question, that how does plant science reduce its carbon footprint? And are both the speakers given great solutions, as Professor Bansal showed us that the solution can begin with at the genetic level, and through precision breeding genome editing, we can build carbon efficiency into the crop itself before it ever reaches to the field. Whereas Dr. Bajgai reminded us that the soil beneath our feet is one of the most powerful tool that we have, and the conservation agriculture is not a future aspiration, but it's a field-ready solution available today. And I hold on to what exactly lies in the essence, that farmers who practice this are themselves are plant scientists. So both presentations are directly directly be applied to the solutions at the field. And also thanks to both speakers of this session, and thanks to my co-moderators, Kavita and Trevor, for keeping this session running so smoothly. A very special thank to Jason and ASPB for hosting this webinar, these Plantae webinar series, and for building a platform where the global plant science community can come together around what matters the most. And as you all know, that the recording is available, will be available on the YouTube channel of Plantae, and also on plantae.org. And we hope to see that you also join us for the next webinar also. So with this, thank you all for joining today's webinar, and thank you to my team, to all the speakers, and thank you so much for joining us today.
Thank you, everyone. We hope to see you in the next one. Have a great week. Bye, everyone.
Thank you. Thank you, all of you. Thank you. It was really great.
Thank
We enjoyed
you.
it.
Thank you.